Hello everyone and welcome to the 7th student. Today we're doing C1.3, which is photosynthesis in IB biology. Let's get started. So what is photosynthesis? Okay, photosynthesis simply is an energy conversion from light energy into chemical energy, which is found in carbon compounds and in this case glucose. So this applies to most of the chemical energy needed for life processes in ecosystems, okay? It all comes from here, or most of it comes from here. So plants can convert CO2 and water into carbohydrates, basically glucose, via photosynthesis. Importantly, in this process, we need hydrogen, and that's obtained by splitting water using light energy. That's called photolysis. So in photolysis, oxygen is a byproduct which diffuses outside photosynthetic cells and eventually into the air through the stomata. Okay, so that's an important side reaction to remember because it's very important in the main reaction. Okay, so let's look at pigments. So photosynthesis requires absorption of light through pigments. We just saw that light is very important, right? So basically, when pigments absorb light, that causes the electrons in the pigment to get excited. They jump to a higher energy level. You'll be familiar with this if you study chemistry. Otherwise, that's all you need to know. A specific energy is required for this to happen, and that energy is only supplied by certain wavelengths. So that's why pigments appear to be a certain color. They are basically the color that they do not absorb, right? Thus, they reflect it. So the wavelengths of light absorbed by a pigment are shown on a graph called an absorption spectrum. So in this case, the x-axis is the wavelength of light. Visible light is 400 to 700 nanometers, and the y-axis is absorption. So for example, chlorophylls, which you can see here in green, are green. Then they are green colored, right? Because the red and blue parts of light excite the electrons, but the green cannot. And you can see that in this absorption spectrum. An action spectrum, on the other hand, is the rate of photosynthesis at each wavelength of light. They're very similar, but don't confuse them because they are slightly different. It is, though, very, very similar to the absorption spectrum, right? As you can see, at blue and red, it has very high rates of photosynthesis. At green, it does not so much, right? So it's very, very similar to the absorption spectrum, but remember the y-axis is a different measurement. Okay, this is a side note, but you need to know how to do this. This was actually in my exam back when I took it in May 2021. So you need to be able to calculate the RF, the distance run moved by pigment over the distance run moved by solvent, right? This is in chromatography experiments. So if you have a diagram like this, you calculate from the baseline to the pigment front and from the baseline to the solvent front, and you can do the division. Super, super easy, but make sure to remember it because it's one of those things that's easy to skip. Great. So now in terms of photosynthesis, what are some limiting factors? Well, first, CO2 concentration and light intensity, obvious ones. Why? Because you need them. You can see it in the equation that you need them, right? Why is the graph like this, right? It's basically you get a plateau at the end. That's because at one point, right, there's enough light intensity that it becomes a limiting factor, right? So at this point, basically, same goes for carbon dioxide, by the way, but at this point, you don't have enough light. So when you increase the light, right, you basically get more photosynthesis happening. At one point, though, there's too much light. There's basically enough light, right, that there's another factor that's limiting. There's maybe not enough CO2 or the temperature's not high enough, right? So even though you can increase the light intensity, that doesn't increase the rate of reaction. Same goes for carbon dioxide. So why does the graph for temperature look different? Okay, because with temperature, we're looking at denaturation. Remember, these processes in photosynthesis require enzymes. And remember, after a certain temperature, enzymes denature and they stop working. So that's why when you increase the temperature to a certain extent, it increases the rate because the enzymes are more active and there's a higher chance of collision. But after a certain while, they all denature. Good. Hope that's clear. Remember limiting factors, a theme that comes up again and again in IB. So try and remember it. Okay. This is again a side note, but it's part of the new syllabus. So, okay, basically in greenhouses, okay, the temperature and light intensity are kept constant normally, but the CO2 can vary. And basically, they found that increasing the CO2 above atmospheric levels has been found to increase rates of photosynthesis. That makes sense, right? So this is a common practice on sunny days. On sunny days, because the light intensity is high enough, right, that it's CO2 that's probably a limiting factor. So this ties in to what we just talked about. If this is not clear, ask in the comments and I'll give you a better answer. But basically, they then expanded this research, right, to the atmosphere. So we know that CO2 levels in the atmosphere are rising because of climate change, so it's possible that actually there will be more photosynthesis and plant biomass will increase. If that happens, right, remember photosynthesis reduces atmospheric carbon dioxide, so it's being tested whether actually this increase in CO2 levels will self-regulate, right, due to an increase in plant photosynthesis. How is this tested? Well, it's tested in free air carbon dioxide enrichment experiments. That's what it's called, right? Basically, they built huge towers which release CO2, and then they measure rates of photosynthesis and biomass production. So this is all for SL. Moving on to HL, now we have to look at the actual intricacies, as always. I'm so sorry for you, but let's go. So first, photosystems, what are they? Okay, so photosystems are complexes made of a molecular array of pigments. So as you can see here, we have an array of pigments, basically, and these are located in the thylakoid membrane of chloroplasts, okay? So normally each photosystem has about 100 chlorophyll molecules and then 30 accessory pigments, such as carotene. So they're very complex. So each photosystem has what's called a core complex, okay? And that's connected to light harvesting antenna complexes. So you can see here the core complex and the light harvesting complexes. How does this work? Okay, so basically pigments within the antenna complexes absorb light because it causes electrons to become excited and jump to a higher energy level. Remember, we talked about that. After excitation, the electrons are going to drop back to their original level, but in doing so, they're going to excite the adjacent pigment, right, molecule. That's called excitation energy transfer. And this is going to happen repeatedly, right? So jump, 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 until it reaches the electrons of a special chlorophyll in the core complex, okay? These are the special chlorophylls in pink. These chlorophylls are able to donate pairs of excited electrons to electron acceptors, and this basically completes the task of the photosystem. Importantly, there are two types of photosystem, photosystem 1 and photosystem 2, which we'll talk about more later. So what are the advantages to having pigment molecules arranged in this array as found in photosystems? Well, first, light photons, when they reach the photosystem, are scattered. So having many pigments close together improves absorbance. Secondly, the range of wavelengths that a pigment can absorb is narrow, but having more than one type of pigment alleviates this. And thirdly, it allows the electron excitation transfer from one pigment to the other. Otherwise, electrons would just drop down and the energy would be lost as fluorescence. Okay, so it makes sense why we have this structure, right? Everything in biology makes sense, even though you might not think so at first. So what happens with these photosystems? Okay, so we have the light-dependent reactions, okay? Photosynthesis is divided into light-dependent and light-independent reactions, and that's a very important distinction to make. So first, we have photosystem 2 at the start here, okay? Remember, this is happening in the thylakoid membrane. So when photosystem 2 absorbs photons, the special chlorophylls are excited, its electrons are excited, right? And then these electrons are basically going to jump between carriers. Does that ring a bell? I hope it does. So that's from cell respiration. Go back and look at that video because they're highly related. So the electrons are going to jump between electron carriers, and that energy is going to be used to pump hydrogen protons into the thylakoid lumen, forming a concentration gradient that then can go down the ATP synthase. I'm speeding through this because, again, it's in cell respiration, but if you do have any questions, leave it in the comments. So then the protons go down the ATP synthase. Remember, this is a motor which can rotate and produce ATP, okay? Importantly, you might think, okay, so if all of these electrons in photosystem 2 are getting excited and leaving, why are they not depleted of electrons? Okay, well, that's a good point. Basically, we need to regenerate those electrons, and that's done through photolysis. Okay, so basically what that means, we talked about it at the start briefly, but it means that water together with light is divided into oxygen, protons, and electrons. So the electrons go to the photosystem 2. The oxygen is a waste product that goes through the stomata, and the hydrogen is used to form this proton gradient as well, okay? So that's important. That's the role of light and of water here, right? That's why this is light dependent. So that's very, very, very important to remember. And actually, this oxygen that's lost through the stomata is very important as well because it's what increased atmospheric oxygen throughout the Earth's history and allowed aerobic respiration to evolve. Okay, so now that we know that, these electrons, what do they do? What are done with? Remember in respiration, we had an electron acceptor at the end. Here we don't. Here what happens is they go to photosystem 1, right? And by the time they're in photosystem 1, they're ready to be excited again. So again, light hits, they're excited, and they again pass through a short chain of electron carriers. In this case, they touch the enzyme NADP reductase, which converts NADP to NADPH. And you might be thinking, isn't it NAD and NADH? Well, yes, okay. It's the same thing, but basically NADP is the molecule that's used in photosynthesis. It has the same role, but it just has a different name, okay? So now we have NADPH and we have ATP for the next steps of photosynthesis. There's a caveat here, okay? Sometimes the NADP supply runs out. Remember, this is a reoccurring topic, happens as well in cell respiration. So if it's all in NADPH, we run out of NADP. So when this happens, actually, excited electrons from photosystem 1 are redirected back, right? And that stops production of NADPH. Instead, what it does is it produces more ATP, right? That's called cyclic phosphorylation, where photosystem 1 actually starts making ATP and not NADPH in an effort to regenerate the stores of NADP+. Okay. Now, let's talk briefly about thylakoids. So thylakoids are found in the stroma of the chloroplast, and they are where the light-dependent reactions we just described occur. So they are specialized in several ways. So obviously, they separate the lumen of the thylakoid from the stroma, right? So that's important. Then that allows the proton gradient to be maintained and the ATP to be synthesized. They also have ATP synthase on the thylakoid membranes, right? Which is important, as we just described. They have a complex which can split water in the lumen through photolysis. Again, very important to regenerate the electrons of photosystem 2. And they have photosystems, okay? So they have all the things needed for the light-dependent reactions. Good. Now let's talk about the light-independent reactions, okay? So the first step is carbon fixation. So once we've already produced NADPH or reduced NADP and ATP, we can do the light-independent reactions. So first, how does carbon actually go into the photosynthetic reaction? Okay, it's called carbon fixation, and it occurs in the stroma of the chloroplast. Basically, CO2 reacts with ribulose biphosphate, and it produces glycerate 3-phosphate. That you do need to remember these names. And the enzyme that catalyzes this is called rubisco. Super important. It's the most abundant enzyme on Earth, even though it's very inefficient. That's why it's the most abundant, because we need a lot of it, because it's not very, very efficient. Okay, so do remember this, okay? It's very, very important. This comes up a lot. This process we just talked about is actually part of a bigger process, which is called the Calvin cycle. So as you can see here, CO2 is converted by rubisco into glycerate 3-phosphate, and then glycerate 3-phosphate is going to be converted into triose phosphate. Okay, you can't see it here, but it's here. Triose phosphate, and that requires NADPH and ATP, which is great because we just produced that in the light-dependent reactions. Triose phosphate now can either be converted into ribulose biphosphate again or into glucose, okay? But importantly, this is a cycle. So ribulose biphosphate needs to be regenerated for the Calvin cycle to continue. And basically, only 1 out of every 6 triose phosphates can be taken out to make glucose, since only 1 out of every 6 carbons comes from CO2 in this cycle. So basically, what can enter can leave, but nothing else, right? Because then every time you would have less in the cycle until you had nothing. So 1 out of 6 goes into making glucose, 5 out of 6 goes into regenerating this ribulose biphosphate. So basically, for a net gain of 1 glucose, the Calvin cycle has to happen 6 times, okay? And this regeneration of ribulose biphosphate requires ATP, as you can see here, right? You can do the numbers to see why it's this 6th and these 5/6 as well, right? If you work it out, you basically need 6 carbons to form a glucose, right? So, but then ribulose biphosphate has 5 carbons, so that's why you also need a 5/6 for every 1/6 that goes to make glucose. So after the Calvin cycle, these glucoses produced are used to make other carbohydrates. So you can make sucrose, which is used to transport to other parts of the plant, starch for storage. This happens when there's very, very high glucose production and you need to store some, right? For maybe the night when there's not enough photosynthesis happening, fatty acids, and even amino acids. So actually all 20 amino acids are synthesized by photosynthesizing organisms, right? Whereas we have to eat some. So basically, all compounds other than glucose are made by metabolic pathways which can be traced back to an intermediate in this cycle, right? This is how plants produce all of their carbon compounds, which is quite amazing, if you ask me. Okay, and then finally, right, In conclusion, we have the light-dependent reactions, which happen in the thylakoid membranes, and they include photolysis, light absorption by generation of excited electrons, transport of electrons by carriers, ATP synthesis by chemiosmosis, and reduction of NADP. And we have light-independent reactions, which happen in the stroma, and they include carbon fixation, synthesis of triose phosphate and other carbon compounds, and regeneration of ribulose biphosphate. Despite their name, actually, light-independent reactions can only go a few seconds in the darkness. Why? Because they depend on ATP and NADPH from the light-dependent reactions, which will run out if not produced continuously. Therefore, these two reactions are interdependent, but one of them is always going to be a limiting factor. So when you have high light intensity, right, carbon fixation is normally the rate limiting step, right? And so actually the light independent reactions, right, is the limiting step. And that prevents photosystem 2 from working since not enough electrons can be supplied due to a lack of NADP. Since producing NADP requires the Calvin cycle, right? And if we're not fixing carbon quickly enough, that's not going to happen. In low light intensity, the production of ATP and NADPH are restricted, right? The light-dependent reactions are restricted. So conversion of glycerate 3-phosphate to triose phosphate is the rate-limiting step because that requires both ATP and NADPH. So that just goes to show these two are necessary and they need to be balanced out so that one doesn't stop. Okay, that's all for the content. Again, I acknowledge this is a very hard topic. So go to cell respiration, watch this over again if you didn't understand it, and if you still have any questions, leave them in the comments. I'm more than happy to respond. And now for some quick questions. So what is used to reduce NADP in the light-dependent reactions of photosynthesis? Okay, in 3, 2, and 1. Okay, it's electrons from photosystem 1. Remember, the electrons go from photosystem 2 all the way to photosystem 1. That's for producing ATP. And then after photosystem 1, they go to NADP reductase, right? And they use the electrons, those electrons, to reduce it, right? Conversion of ATP into ADP, that just doesn't happen there. They don't use protons from the thylakoid space, right? They use them from the stroma, and they don't use oxygen released by photolysis of water. That's used for regeneration of electrons for photosystem 2. Okay, next question. Which processes release the carbon dioxide fixed by photosynthesis? Again, pause, and 3, 2, and 1. 1 and 2. Why? Okay, well, respiration we've seen clearly, right, that it releases carbon dioxide. You can go back to that video. Combustion releases because we're burning basically organic matter, right? And then fossilization actually stores carbon in it, right? That's why we have fossils. So that doesn't release it, actually. It just stores it. Okay. And finally, which of the following occur during cyclic photophosphorylation? We just talked about that. So in 3, 2, and 1. 2 and 3. Why? Okay. We don't reduce an ATP+. That's the whole point of cyclic photophosphorylation, right? It's when we do not want to get rid of NADP because we don't have enough. But we do produce ATP. Remember, the electrons go back from photosystem 1 to try and produce more ATP to balance it out and not have too little NADP+. And then photoactivation photosystem 1 still happens. Those electrons are still excited, but again, they just return back to make more ATP rather than making NADPH. Okay, so that's the end of this video. I know it's tricky. Hope that was clear. Again, any questions in the comments, and I'll see you next week for the next video. Bye, everyone.